today we discuss about the biochemistry. In biochemistry we discuss about the DNA. Okay, deoxyribonucleic acids. What are the building block of nucleic acids? What is the building block of DNA? What is ribose sugar? Like that, we discuss all this in this DNA topic. Okay. First of all, what is the DNA? And what is the full form of the DNA? DNA is the deoxyribonucleic acid. What? Deoxyribonucleic acid. From name, it is clear that this is a deoxy. Means they don't have oxygen atom at the 2' in the ribose sugar. See, this is the ribose sugar, right? This is the ribose sugar. 1', 2', 3', 4', and here is 5, right? This is the ribose sugar. Each prime having OH group and HI. OH group, right? So, in DNA, these 2' don't having OH group. This is only hydrogen atom will be present here. So, this name is come from that. Means there is deoxy. Means there is no oxygen. So, this is deoxyribose nucleic acid. What? Nucleic acid. Means nitrogenous base these are acidic in nature. What? DNA is in acidic nature. From name it is clear that DNA is what nature? This is the acidic in nature. The chemical substance is present in the nucleus, all cells in all living organisms. It is either eukaryotes or it is either prokaryotes, both having a DNA. Either this genetic DNA is actually called as the genetic material. What? Genetic material. Because DNA contains all the information of the cells. Here, but in this chapter, in this biochemistry, we discuss only about the biochemical nature of the DNA, not genetic nature. Okay. So, DNA are present in all eukaryotes or prokaryotes. In prokaryotes, DNA are float in cytoplasm, whereas in eukaryotes, they having a outer membrane that is called nuclear membrane. They are present in nuclear membrane, or they are present in the nucleus. DNA control all the chemical changes which take place in the cell, right? All the chemical changes, all the information that are encoded for protein or RNA, that all are controlled by this DNA molecule. This DNA molecule, that is deoxyribonucleic acids. The kind of cells which is formed is controlled by DNA. Means which type of cells will be formed in our body, like muscle cells will be formed, or blood cell, nerve cell, which type of cells, which type of enzymes, which type of protein, that all are controlled by DNA. That all are controlled by DNA, because DNA code the sequence or information for all the formation of the cells. The kind of organism which is produced is controlled by DNA, like buttercup, giraffe, here human, every kind of organism. Every kind of organism are controlled by DNA. only and only the element that which control the whole organism, whole the structure of organism, that is the DNA. What are the molecules are present in the DNA? See, DNA is a very large molecule that is made up of long chain of subunit. Long chain of subunit. The subunit are nucleotides. The subunits are nucleotides, right? Subunits are nucleotides. Each nucleotide is made up of deoxyribose and a phosphate group and an organic base. See here. This is the structure of ribose, that is deoxyribose, right? Okay, this is the structure of deoxyribose. Deoxyribose, clear? A phosphate group are present here. Okay, this is the phosphate group are present here, and the organic base. Organic base are present here. This is the nitrogenous base. What? This is the nitrogenous base, that is maybe adenine, cytosine, guanine or thymine. Okay? This nitrogenous base are maybe adenine, cytosine, guanine or thymine. So this is the structure of the DNA. Not DNA, this is the molecule by which this chain of the DNA is formed. Now by this nitrogenous base are attached by phosphate group, another like this, and here hydrogen bond will form. Okay, clear? So DNA is a very large molecule made up of a long chain of subunits, that is nucleotides. This is called as nucleotide. What? This whole structure is known as nucleotide. And this nucleotide is made up of phosphate group, deoxyribose sugar and organic base. Right? How chain will be formed? This phosphate group react with another hydroxyl, this hydroxyl group of another ribose sugar, and like this polymer of chain will be formed. And this nitrogenous base will form hydrogen bond with in parallel of their this another DNA molecule. Right? Clear? So ribose and deoxyribose. Ribose and deoxyribose. Ribose is a sugar like glucose. Ribose is nothing but is a sugar like glucose, but with only 5 carbon atom in its molecule. This is a 5 carbon atom. 1, 2, 3, 4, 5. Right? Deoxyribose is almost the same but lacks 1 oxygen atom. Deoxyribose what? It is nothing. Already I told it is almost the same but lacks one oxygen atom at 2' end. What? At 2' end. Both molecules may be represented by the symbol. This is a symbol. This is a 5 carbon atom, that is ribose, but during formation of polymer, this carbon atom will come here, that is C, and here phosphate group will be attached here, right? Phosphate group will be attached here, or this carbon atom will be come here. Okay. Now, what is nucleotide? Nucleotide is the energy-rich component that derive metabolic process in cell. Serve as chemical signals. What? Nucleotides serve as a chemical signals. What is nucleotide? Nucleotide serve as a chemical signal or is a key link in cellular system that transport to hormones and other extracellular stimuli. A structural component of no enzyme cofactor metabolic intermediate. Each nucleotide is formed by 3 units: phosphate, sugar and nitrogenous base. Phosphate, sugar and nitrogenous base. Right? There are 4 type of nitrogenous bases. Actually, there are 2 type of nitrogenous base. Nitrogenous base are of 2 types: purine and pyrimidine. What? Purine and pyrimidine. Purine and pyrimidine. This is purine ring and this is pyrimidine ring. Right? Purine ring is made up of 2 chains and pyrimidine having single chain. Okay. This is the pyrimidine ring, whereas purine ring having 2 rings. This is a purine ring. In pyrimidine, three nitrogenous bases are present. That is cytosine, uracil, thymine. Whereas purine, 2 nitrogenous bases are present. That is guanine and adenine. What? Guanine and adenine. Right? This 3 type of pyrimidine. In this 3 type of pyrimidine, in DNA, two pyrimidine are present. That is cytosine and thymine. What? That is cytosine and thymine. Whereas in purine, both are present in DNA or RNA. In RNA molecule, uracil and cytosine are present. Uracil and cytosine are present. Okay. So uracil are not present in DNA and thymine are not present in RNA molecules. Instead of thymine, uracil are present in RNA. Instead of uracil, thymine are present in DNA. Whereas both guanine and adenine are present in both DNA and RNA molecule. Okay. Remember this structure. This structure is very important. Okay. Now, see here. First is this is a structure of DNA or ribose sugar in RNA. Okay. In DNA or actually DNA, what is the difference between DNA and RNA? These both structure are same. Only are different in a structure. Only difference at this 2' end. In DNA, it is having only H group, hydrogen atom in DNA. Okay. Whereas in RNA, here having OH group in RNA. Only difference is here, only and only. And another difference is in nitrogenous base. That is DNA having thymine and RNA having uracil. Okay. Due to this OH group that is present in RNA, make RNA more reactive as compared to DNA. So DNA is considered as the genetic material of the cell, because DNA is less reactive compared to RNA, because here only hydrogen atom is present that not react with another molecule, whereas this hydroxyl group react with another molecule. Right? So DNA is less stable compared to RNA. Understand? It's clear? What is the difference between DNA and RNA? This is the sugar, that is ribose sugar. This ribose sugar having phosphate group, and here having nitrogenous base, that is organic molecule, and here is H atom or OH in RNA or H in DNA, and here it is OH, hydroxyl group, 3' end. Right? and this is the cytosine. This is the structure, how this link this cytosine structure, right? This cytosine, this is the carbon atom. This carbon react with this nitrogen atom and ring of this nitrogen atom and form a bond. Like that only all the nitrogenous base attached with this carbon molecule at 1 prime, right? This is the most common organic bases. Are what type of organic bases? There are 4 type of organic bases. That is, first adenine, second thymine, third cytosine, and fourth is guanine. Adenine is coded by A, thymine is coded by T, cytosine is by C, and guanine is by G. And RNA, there is another, that is U, coded by U, uracil. Uracil is coded by U. Okay. These are the 4 nitrogenous bases or organic bases. This is nucleotide. What is nucleotide? When nitrogenous bases attached with ribose sugar, and that ribose sugar having also a phosphate group, then this all molecule or this all structure is called as nitrogenous base. What? Remember this. There are so many confusion. What is nitrogenous base? What is nucleotide? What is organic base? This all are more confusing. Okay. So clearly understand what are nitrogenous base. What is nucleotide? What is nucleotide? Understand what is nucleotide. Remember, if tide will be attached, means phosphate group is be present. The deoxyribose, the phosphate and one of the bases combine to form a nucleotide. What? They all combine to form a nucleotide. Remember, phosphate group are present in this. What was present in nucleotide? A deoxyribose, phosphate, and one of the bases. One of the bases means from adenine, guanine, cytosine, thymine. From these all 4 one organic bases are present. One organic bases are present in this, attached with this deoxyribose. Then that is whole structure we call as nucleotide. This whole structure we called as nucleotide. Okay. And what is nitrogenous bases? If only adenine, guanine, cytosine, these are called nitrogenous bases. What is nucleosides? Another is, what is nucleoside? Nucleosides means this phosphate group are not attached. Only deoxyribose sugar and this adenine will attach, then this is called nucleoside. What? Nucleosides. And if this all are attached, then it is called nucleotide. Okay? Clear? What is nucleotide and what is nucleoside? Now, next is how these nucleotides are joined. A molecule of DNA is formed by millions of nucleotides that join together in a long chain. Like you already know that how the protein will join with each other. Protein is a long chain of amino acids. Like that only, DNA is the polymer of nucleotide. What? DNA is the polymer of nucleotide. They are joined in a long chain. See here, this all are what? This all are nucleotide. This single chain are what? These are nucleotide. This nucleotide joined by phosphate and OH group of second prime. Means this is the 5' end and this is the second prime end. This is the 5' end and this is the second prime end. So, this is the 3' end. This is the 3' end. So, always bond formation will be takes place at the 5' end and between 3' end. Okay? Ribose sugar of the 5' and 3'. 5' of the phosphate react with 5' of the phosphate react with 3' of the OH group of the nucleotide. Okay? And form a bond. What? Form a bond. That is called phosphodiester bond. What? Phosphodiester bond. This bond is called form a bond, that is phosphodiester bond. And this bond, after this bond will be formed in between, and this like that only this bond will be formed, and a long chain of nucleotide will be arranged. And this long chain of the nucleotide is the sequence of the DNA. What? This is called as a polymer of the nucleotide, and this is a DNA structure. And this is here what? Here is a base A, T, C, G. One of these base will be present here. Either A, either T, either G. One from this all. Okay? So, polymer will be formed at the, so what happens? If here, if here phosphate group is present, then it is known as 5' end, and if here OH group is present, then it is known as 3' end. So, DNA having end, that is a 5' end and a 3' end. What? 5' end and 3' end. This is called as 5' end and 3' end. Like in protein, there is a N terminal and C terminal. Here in DNA, there is a 5' and 3'. If hydroxyl group are present in last, that is the 3' end. And if phosphate group are present in last, that is known as 5' end. So remember this. Where is the 5' end will become, and where 3' end will become. This is a sugar phosphate. This is called as sugar phosphate backbone. What is this? This is a sugar phosphate backbone. Sugar phosphate means this sugar and phosphate will be occur, and this is a back. So this is known as backbone. In fact, the DNA usually consists of double strand of nucleotide. Generally, DNA is of double stranded, like this. Like this, okay. Sorry. Okay. The DNA is actually made up of two strands. One strand is of 5' to 3' end. Another is 3' to 5' end. If there is opposite prime is present, then that is called as, if opposite prime is present, then that is known as anti-parallel strand. And if same type of prime is present at both ends, that is called parallel strand. Okay. See, this is the 3' end, this is the 5' end. Here also there is 3' end and here also 5' end. This is known as parallel strand. What? Parallel strand. Okay. If here 5' end, here 3' end, and here 3' end, and here 5' end, this is known as 5' end. Here also there is 3' end, and here also 5' end. This is known as parallel strand. What? Parallel strand. Okay. If here 5' end, here 3' end, and here 3' end, and here 5' end, this is known as anti-parallel strand. Okay. Generally, the DNA which occur in our as genetic material or in our body, that is anti-parallel in nature. What? That is anti-parallel in nature. The DNA that occur in our body, that are in anti-parallel in nature. That is one side 3' end, then another side having 5' end. The DNA usually consists of a double-stranded of nucleotide. The sugar phosphate chain are on the outside and the strand are held together by chemical bond between the bases. Okay. And bonds are between the bases. What type of bond are present in between the bases? That is A, T, G, C. What type of bond are present in this? The bond present in this nitrogenous bases or organic bases are the hydrogen bond. What? Hydrogen bond. If A will, another thing is what? Always purine. Purine will combine with pyrimidine. Okay? Pyrimidine. Purine will combine with pyrimidine. Okay? What happened? Purine combined with pyrimidine. Purine combined with pyrimidine. And there are specific base which combine with a specific base. A specific base of purine is combined with a specific base of pyrimidine. Not all purine bases will combine with all pyrimidine bases. Means any of the purine bases will combine with any of the pyrimidine bases. This does not occur. Means adenine combined with thymine. Adenine combined with thymine. Guanine combined with cytosine. Okay. And adenine when combined with thymine, they form double hydrogen bond. And when guanine combined with cytosine, they form triple hydrogen bond. So guanine and cytosine are more stable compared to adenine and thymine. Okay. So this line is clear now. The sugar phosphate chain are on the outside, that is the sugar phosphate chain are present as a backbone. What? They are present as a backbone of the DNA. And the strands are held together by a chemical bond that are between the bases. Means that are by the hydrogen bond that are present in between the bases, that is AT or CG. Okay. Clear it is? See, these are the two strand DNA. This is the first strand of the DNA, and how the other strand will come and combine with this strand. If there is A, if there is A, then here T thymine bases will come and form bond with A. Not G base will come and form with adenine. No, never. Only thymine will come and bind with adenine. Okay. If their G will be present, then your cytosine will come and form 3 hydrogen bond with guanine. Okay. And if here cytosine is present, then here guanine will come. Guanine base will become, nitrogenous base will come and combine this. And if here thymine will be present, then here adenine will become and combine with 2 hydrogen bond. Okay. Remember this. Only specific, they are specific for each other. If A is present, then T will become, and if G is present, then C will become. Okay. Nothing else will come and bind with this. Okay. Remember this. Most important this is. Okay. And uracil is not present in DNA. If here RNA is present, 2 strand of RNA, then here uracil will be present. Okay. See here, how the bonding will be formed. The bases always pair in the same way. Adenine form a bond with thymine. Adenine bonds thymine, that is by 2 hydrogen bond, and cytosine bond with guanine. Already I told you, by 3 hydrogen bond. Okay, understand this slide is clear. Now this is how bond formation will takes place. Bonding 2, how bond formation will takes place. Same it is. This is one strand. This is the one strand of the DNA. This is one strand of the DNA. This is the second strand of the DNA. Here it is 5' end. Here it is 3' end. Here also there is 5' end. Here also 3' end. Okay, means they are parallel in nature. They are parallel in nature. Huh? And both bases, nitrogenous base will came and combine with their complementary bases, and bond formation will be takes place. Okay. Now the pair strand are coiled into a spiral called a double helix. How the double helix DNA will be formed? Actually, when this nitrogenous bases will be formed, then there is helical formation will be takes place. Like alpha helix, you understand in protein structure, like that only. There is a helical form will be takes place. That form, because hydrogen bond formation will takes place. During the formation of this hydrogen bond, there is a helical pattern of the DNA will be takes place. This is known as double helix DNA. What? This is known as double helix DNA. This is a sugar phosphate backbone. This is a backbone, and this is a nitrogenous basis by which DNA will be formed. Okay. This sugar phosphate chain provide support or stability to the DNA. Provide stability to the DNA. This sugar phosphate bond. Okay. Now this replication topic we cover in genetics. In briefly, I told you what is replication. Replication is a type of division, type of increase the number of itself. Actually, it is a itself copy. Increase the number of itself copy. We Before a cell division, the DNA strand unwind and separate. Each strand make a new partner by adding a appropriate nucleotide. The result is that there are now two double-stranded DNA molecules in the nucleus. So that when the cell divide, each nucleus contain identical copy. What is replication? Actually, what is replication? Nothing but it is a copy. Increase the copy number. Increase the copy number, or it is the increase the duplicacy or duplicate their number. Okay. In genetics, we study more about this replication topic. Actually, we discuss about the one type of question generally arise. Which of the following repeats a correct pairing of bases? Because we already in starting we discussed that types of base, adenine combined with thymine, guanine combined with cytosine, like that only. Okay. So what happened here? Which are the correct bases? Adenine combined with thymine, right? This is correct. This is correct. Adenine combined with thymine and adenine combined with guanine? No. Thymine combined with adenine? No. Guanine combined with cytosine? Right. Thymine combined with thymine? No. So adenine combined with thymine and guanine combined with cytosine. This is the correct pairing of the bases. Okay. So this is all about the DNA and something about RNA. Okay, understand? So today we stop here. The DNA topic will be complete. So today we discuss mainly about the digestion of nucleic acid, how the nucleic acid are digested in our body, right? So mainly there are two pathways, that is the salvage pathway and de novo pathway, by which the nucleic acid are digested, right? So first of all, what are the nucleic acids, right? What are the nucleic acids? So already you studied that the DNA and RNA, right? Ribonucleic acid and deoxyribonucleic acid. Already you discussed this, right? So this DNA and RNA are the two types of nucleic acids that are present in our body, right? This is the genetic material. Also DNA is the genetic material, right? DNA is also the genetic material. And this both DNA and RNA both are made up of purine and pyrimidines. The difference was what? The presence and absence of hydroxyl group at the 2'. What? the presence and absence of the hydroxyl group at the 2' of sugar phosphate bond. Right? In deoxyribonucleic acid, from name it is clear that it deoxy. Right? From name it's clear, it is the deoxy. Means absence of the 2' hydroxyl group. And in RNA, that 2' hydroxyl group at the sugar molecule, there are presence of the 2' hydroxyl group, right? And another difference was the presence of uracil. Presence of uracil are present in the RNA, and thymine are present in the DNA. Clear? This is the main difference in the DNA and RNA. So now we discuss in detail how this nucleic acid are digested. So what are the nucleic acid? Already I told you that genetic information that is passed are with the help of DNA or RNA. Clear? It is with the help of DNA or RNA. And this DNA and RNA are found in all organisms. Clear? In all organisms. Either in a small or in microscopic organism, mainly it is either DNA present or either RNA or present. But one genetic material are always present, right? Remember this. So this is found in all food sources that are from animal and plants, DNA and RNA. So how this nucleic acid are break? We eat some food, like any type of food, right? It is the plant food or animal food, any type of food we eat that contain nucleic acid. Then how that nucleic acid are digested in our body, right? So the first step of digestion was the mastication, means mechanical digestion, right? And propulsion, that is swallowing, bolus of the food in the orophagus or esophagus, right? See, we eat the food, we chewing like this, right? This is also a part of break down the food, right? Then we swallow that food, right? That is the bolus of the food. So from mouth to stomach, we eat this food, they convert it into the bolus of the food, and that bolus of the food go in the esophagus, right? So now esophagus to stomach. By peristalsis, bolus of food is propelled down through the esophagus toward the stomach, right? This bolus of the food are propelled down and they are go toward the stomach, right? This is the bolus of the food. This is longitudinal muscles contract, shortening, and passage. They make a passage ahead of the bolus, and this is the gastropharyngeal sphincter closed, and the circular muscles contract and constricting passageway and pushing bolus down. Clear? So now small intestine. The food are go in the small intestine. So most of nucleic acid that are eaten, the most of the nucleic acid digestion takes place within the small intestine. Clear? Most of the nucleic acid are digested in the small intestine, and nucleic acid are dissolved in gastric chyme, which is deposited into the duodenum, and pancreatic juice, which contain nucleases, is released to initiate nucleic acid degradation, right? Is released to initiate nucleic acid degradation. So this is the nucleic acid, right? This is the Hind3, and this Hind3 is the restriction enzyme. What? This is the restriction enzyme, and they broken down the strand, right? So nucleases are enzymes that cleave the chain in nucleotide in nucleic acid into smaller units. They cleave into the smaller units. So activity of nucleases, there are two types of nucleases, endonucleases or exonucleases. Clear? There are two types of nucleases, endo and exonucleases. Intimal cuts, nucleotide removed from the ends. Now absorption. So nucleotides undergo further degradation in the microvilli. The nucleotide are further degradation of the nucleotides will go in the microvilli, and their further degradation will be takes place. This is the microvilli. Clear? So enzyme phosphatase and nucleosidase, right? Like enzyme phosphatase and nucleosidase into three constituent parts, and membrane transport proteins bring product across basolateral membrane into capillaries, right? Into the capillary. So this is the villi, right? This is the blood vessels in folds of intestine. This is the having the blood vessels, right? In the fold of intestine, and this is the villi, right? And this is the microvilli, right? Where the further digestion of nucleotides will be takes place. Clear? Further digestion of nucleotides will be takes place here. Clear? So what happened? This is the brush border cells, and this is the capillaries. So now nucleotide. It is the nucleotidase. It is the from ase. Ase is the specific term that occur when this is, suppose this is nucleotide, right? And ase will be joined with this, right? Nucleotidase means this is the enzyme. Ase represents the enzyme. Clear? So here what happened? Nucleotidase means it is the enzyme. So it is the hydrolytic enzymes that catalyzes the hydrolysis of a nucleotide. Hydrolysis of the nucleotide into a nucleosides and a phosphate. Into a nucleosides and phosphate. Clear? So nucleoside is the base plus sugar, right? Nucleoside is the base plus sugar, and nucleotide is the base plus sugar plus phosphate. Clear? From name C. This is the side. S is present here, means S represent the sugar, right? Here T, what represent? Phosphate. T are present in phosphate, means this is phosphate are present. So nucleoside is made up of base and sugar. Nucleotide is made up of base, sugar and phosphate. Clear? Base, sugar and phosphate. Clear this? Clear this all, right? This is the base. This hexagonal ring are the base, and this pentameric ring are the sugar. So this hexameric are the base, and this is the sugar. And when this phosphate are attached to this base and sugar, they are known as nucleotide. So overview of the degradation of the nucleotide is the nucleoprotein are converted into nucleic acids, then they are converted into nucleotide. The nucleotide are converted into phosphate plus nucleosides, and then phosphate plus nucleosides are converted into free purine or pyrimidine base or ribose or deoxyribose. Clear? Because nucleosides, right? Nucleosides means they contain nucleotides and the base. Nucleosides means they contain nucleotide and the base. And the nucleotide are made up of the purine or pyrimidine. What? Nucleotides are made up of purine and pyrimidine. So we have to break the nucleotides into purine and pyrimidine. Right? After that, this purine and pyrimidine are converted to cytosine, adenine, cytosine, guanine, or uracil. Right? So, ribose or deoxyribose is also a type of sugar. They are converted into deoxyribose, means when totally the digestion of the nucleic acid will be occur, means there are presence of the purine or pyrimidine, means adenine, guanine, cytosine, uracil, thymine, right? Either one of them, right, are present, or ribose sugar are present, or deoxyribose sugar are present.
if their digestion of the DNA will be takes place, means deoxyribose sugar are present, and if digestion of RNA will be takes place, means ribose sugar are present. Clear? So nucleoprotein are digested into nucleic acids. After nucleic acids are digested into nucleotide, and then nucleotide are digested into phosphate plus nucleosides, and nucleosides are digested into nuc- nuc- nucleosides are digested into purine, pyrimidine plus sugar base. Right? Clear this? So overview of degradation are this is the picture, right? So this is the double-stranded chromosome having 6 bases pair. Having 6 bases, 1, 2, 3, 4, 5, 6. They're having a 6 bases pair, right? And chromosome are snipped apart by nucleases in the duodenum by the small intestine. And these 6 bases sugar are go into a duodenum via the small intestine. So these enzymes are secreted along with the pancreatic juice and differ depending on the type of nucleic acid. The work on and the spot on the nucleic acid that they cut apart. So nucleases break apart the hydrogen bonds in the chromosome into individual nucleotides. So in the brush border nucleotide, means brush border nucleotides are then made by phosphatase, right? They meet by the phosphatase, right? An enzyme which separates the phosphate from the molecule, making it a nucleoside. So nucleosides is further degraded by nucleosidases, which separates the pentose sugar from the nitrogenous base. From the nitrogenous base. So membrane transport protein bring these components across the membrane and into capillary of the small intestine. Clear? This is the 6 base sugar. They having a thymine, cytosine, adenine, thymine, guanine, cytosine, right? They having nucleotide, phosphate group, right? And this when they broken down, they goes into a small intestine. Then after that, they duodenum, right? So this is the purine. This is the pyrimidine. Purine are the adenine, guanine. Purine are the adenine and guanine, and pyrimidine are the cytosine, thymine, uracil, right? Cytosine, thymine, uracil. Remember this already I told you that thymine are present only the DNA and uracil are present in the RNA. Clear? So break down into ammonia and beta amino acids, and this purine break down into uric acid. Clear? Means adenine and guanine are broken down into the uric acid, whereas cytosine, thymine, uracils are broken down into the beta amino acids, broken down into ammonia, carbon dioxide, or beta amino acids. Clear? So Salvage pathway versus de novo synthesis. This is the two pathway by which there is broken down of the nucleic acid will be takes place. So free purine and pyrimidine. Free purine and pyrimidine bases are in large part salvaged and reused to make nucleotide. What happened here? They reused to make nucleotide from degradation of DNA and RNA. So they consume less energy. Than de novo synthesis, and is energetically the preferred source of nucleotide for the nucleic synthesis. Clear? What happened here? In Salvage pathway, what happened? Free purine and pyrimidine bases that are present in large part that are reused to make nucleotide. That purine and pyrimidine are reused. What? They reused from the degradation of the DNA and RNA, and they are used to make again, right? So they consume less energy than de novo. Salvage pathway, what happened in salvage pathway? Salvage pathway contain less energy, right, than de novo synthesis, and is energetically the preferred source of nucleotides for nucleic synthesis. So what happened here? They occur in cytosol of the liver, small intestine, and thymus. Clear? and from a small molecule readily available in cell, right? So ribose 5-phosphate CO2 ammonia is the end product. This is the nucleic acid this is a nucleotide biosynthesis, right? This is the de novo, this is the salvage, right? What happened? Purine, pyrimidine, IMP, UMP, right? Here what happened? AMP, they are converted into ATP, CMP, they are converted into CTP, UMP converted into UTP, and then CTP, then deoxy TTP. Where salvage, what happened in salvage? Salvage utilize their purine and pyrimidine, means they utilize the degraded purine and pyrimidine. That's why they require less energy as compared to de novo pathway. Clear this all, right? So now uric acid, hyperuricemia and gout. Now what are the diseases what are the diseases that are caused due to the presence of extra amount of nucleic acid or less amount of nucleic acid, or extra amount of the, like when degradation will take place of the nucleic acid, they convert it into uric acid, right? Already I told you, when degradation of the nucleic acid will be, or digestion of the nucleic acid will be takes place, they convert it into uric acid, and that uric acid are collected if that uric acid are collected in a knee, right? Then they cause a type of disease. Or if uric acids are collected in the joints, they cause a disease like a gout, right? Gout. So uric acid, that is urate, is the end product of urine degradation in humans. Already you see that in the flow diagram, right? Means purine are degraded into the uric acid, right? So hyperglycemia is a serum urate concentrate in excess of urate solubility, that is 6.8 mg per dl, right? 6.8 mg per dl. So is a common serum abnormality, but does not result in gout without crystal deposition. So gout is the disease that is still resulting from deposition of monosodium urate crystal in tissue. Clear? So review of the hyperuricemia cascade. What happened here? This is the dietary purine, tissue nucleic acid, endogenous purine synthesis. Here they converted into urate. Then overproduction of the urate will, if overproduction of urate will be takes this, what happened? They cause as a hyperuricemia. What? Hyperuricemia. And this hyperuricemia having a different type of sign, right? Like silent tissue deposition, gout, renal manifestation, or associated cardiovascular events and mortality. So this is the symptoms of the hyperuricemia, or different type of disease. What type of diseases are caused the hyperuricemia, like silent tissue deposition, gout, renal manifestation, associated cardiovascular events, and mortality. So hyperuricemia cascade, evolution of hyperuricemia and gout. Over time, untreated chronic hyperuricemia increases body urate stores, advancing the severity of the disease, right? See here what happened. So now gout disease. What is the gout disease? See, mainly the gout disease are caused at the joint, like this joint, elbow joints, knee joints, right? So at every point there, if there is deposition of the uric acid will be takes this, then they cause a gout disease. Clear? So this is the knee joint, right? Knee joint. If uric acid are deposited in this area, then they cause a gout. Ankle joints, right? Big toe, right? Wrist, elbow, finger, means at every point of the joints, if uric acid are deposited, means crystal of uric acid are deposited, then they cause a gout. Now, now the drug allopurinol is prescribed to individuals suffering from gout disease. It work it work by inhibiting the oxidation of xanthine. It work by inhibiting the oxidation of xanthine, means the precursor to uric acids. They inhibit the xanthine, means if they inhibit the xanthine, then their production of uric acids will be inhibited, right? So regulation of uric acid is an effective way to inhibit the gout proliferation. Clear? So now defecation. So combination of intrinsic and parasympathetic defecation reflexes. Second, at convenience, while salva maneuver, closure of glottis, deep inspiration, abdominal contraction. So inhibition of external sphincter via ependymal nerve. This is a voluntary control. So now what happened? The defecation deflection, a series of impulses that trigger the release of feces. So sensory fiber sense stretches of rectum wall due to filling, and parasympathetic fiber trigger contraction of rectum wall and relaxation of internal and sphincter. So external anal sphincter can allow defecation or voluntary contract to temporarily stop the reflection, right? So intrinsic defecation reflex. Stimulus, receptor, afferent center, afferent effectors, and response, right? So parasympathetic defecation reflex. In this also, feces enter the rectum, distension of rectal wall, stretched receptors in the rectal wall, sensory fiber terminating S2, S4 sphincter cord segments. So this all about the parasympathetic defecation reflex, right? So sitting posture and squatting posture. Now, valsalve maneuver. Forced expiration with mouth and nose closed. Otoscopy show lateral bulging of the tympanic membrane. So it increases intrathoracic pressure to as much as 80 mm Hg. So this all about the nucleic acid digestion. So thank you.